We described the common mechanisms of bacterial resistance to antibiotics. Now let's explain the mechanism of action and spectrum of activity of beta-lactam antibiotics. There are four classes of beta-lactams. There are penicillins, cephalosporins, carbapenems, and monobactams. And they are all bactericidal. What bactericidal means is that they actually kill the bacteria very fast. So as time goes forward, the number of colonies actually drop very quickly. As opposed to bacteriostatic agents that don't necessarily drop the number of colonies, but actually halt the growth of bacteria. So the number of colonies actually stay the same over time. Beta-lactams bind to penicillin-binding proteins, or PBP. And there are different types of penicillin-binding proteins which result in different spectrum of activity of beta-lactam antibiotics. Now, the first beta-lactams are penicillins, so these enzymes were named after the first beta-lactams, so they are called penicillin-binding protein, but they are also known as transpeptidase. And transpeptidase is an important part of cell wall synthesis. So by inhibiting penicillin-binding protein or transpeptidase, beta-lactams inhibit cell wall synthesis. Now, without going into too much detail, the synthesis of peptidoglycan involves several steps, and one crucial step is the cross-linking of components of peptidoglycan, and that cross-linking is done by transpeptidase enzymes, and that is the site of action of beta-lactams. Let's take a look at the spectrum of activity of penicillins and monobactams first. So when it comes to penicillins, these are they're essentially subclasses of penicillins. So the first penicillins to come to the markets were actually called penicillin, and we have two different forms, penicillin G and penicillin V, with identical activity. And these have good activity against beta hemolytic strep, but they may not have the best activity against Streptococcus pneumoniae. Now, they are active against most E. faecalis, but they have no activity against Enterococcus faecium, and they are not active against any Staphylococcus aureus. So MSSA and MRSA are not active. There is a small subset of Staphylococcus aureus called penicillin susceptible Staph aureus that these agents are active against, but those are essentially less than 10% of all Staphylococcus aureus. And the reason is that majority of Staphylococcus aureus actually produce penicillinases that break down penicillin, so they don't have activity against MSSA or MRSA. And they don't have any activity against gram-negatives as well as anaerobes or atypical organisms. Now, the next set of penicillins are referred to as anti-staphylococcal penicillins. So these are nafcillin, oxacillin, dicloxacillin. So these are colored the same just to help you navigate this chart. So the three have identical spectrum of activity. Now, they are called anti-staphylococcal penicillins because they have activity against MSSA. However, they are not active against MRSA. In fact, none of the penicillins are active against MRSA. Now, when these became stable against MSSA, it came at the cost of losing some of the activity, including they lost activity against E. faecalis, and they had generally poor activity against beta hemolytic strep and strep pneumo. Now, the next set is aminopenicillin. So this is ampicillin and amoxicillin, which have identical spectrum of coverage. Ampicillin and amoxicillin, they do have improved gram-negative activity compared to the previous subclasses of penicillin. So they get some activity against H. flu and including E. coli, Klebsiella, and protease. But it comes at a cost because they lost activity against MSSA because they are no longer active or stable against penicillinases that Staph aureus makes. Now, they do have activity against beta hemolytic strep and E. faecalis and somewhat against strep pneumo. Now, by adding beta-lactamase inhibitors to these two, so now if you combine ampicillin to sulbactam and amoxicillin with clavulanate, you gain some of these activity. So now the penicillinases from Staphylococcus aureus are neutralized by these beta-lactamase inhibitors. So ampicillin, sulbactam, and amoxicillin clavulinate are active against MSSA, and they do gain better activity for gram-negative organisms. And more importantly, they also gain activity against Bacteroides, which are anaerobic organisms. Okay, so Bacteroides also make penicillinases that pretty much kill majority of penicillins, but the ones that have beta-lactamase inhibitor, including piperacillin-tazobactam, they gain activity against Bacteroides. Now, Bacteroides become very important when we talk about intra-abdominal infections. Now, the last and the broadest penicillin is piperacillin-tazobactam, so excellent activity against beta hemolytic strep, against strep pneumo, as well as E. faecalis, but not against E. faecium. It does have activity against MSSA, but not MRSA, and pretty good gram-negative coverage overall. But more importantly, this is the only the only penicillin that has activity against Pseudomonas aeruginosa. Now, the only monobactam on the market is Aztreonam, and it's important to note that Aztreonam does not have activity against any gram-positive organism, so it's all red for gram-positives. Now, it does have some good activity for some of the gram-negatives, including H. flu, as well as some of the Enterobacterialis. However, it does have somewhat poor activity against Pseudomonas. So over time, activity of Aztreonam over Pseudomonas has decreased, and moreover, it doesn't have activity against Bacteroides or atypicals. Now, let's take a look at cephalosporins. We have a lot of cephalosporins on the market. These are select few that are more commonly encountered, but there are a lot more. And cephalosporins in general are thought of in generations. So there is first generation, second generation, third generation, and fourth generation. And then at some point, we kind of stopped calling generations because it started to be inconsistent. Now, in general, you go from low, narrow spectrum to broad spectrum as you go higher with the generations. So we have for the first generation, cefazolin and cephalexin. They have essentially identical spectrum of coverage, so good activity against beta hemolytic, somewhat activity against strep pneumo, and nothing against Enterococcus species. And they do have activity against MSSA, so even though Staphylococcus aureus makes penicillinases, it cannot break down cephalosporin, so this is active, but they do not have activity against MRSA. In fact, ceftaroline is the only beta-lactam in general that has activity against MRSA in the US among the agents that are available in the US. There are other beta-lactams that are available elsewhere that may have activity against MRSA. Now, for gram-negative, first generation, they have somewhat activity against E. coli, Klebsiella, and Protease, but you typically need susceptibility results to confirm, and they don't have activity against Pseudomonas, Bacteroides, or atypicals. Now, when we go to second generation, again, it's important to note that there is a subset of second generation referred to as cefamycin, so that's cefoxitin and cefotetan. And the rest of second generations, one example is cefuroxime. Cefuroxime has good activity against beta hemolytic, but not strep pneumo. And again, all of them are good with MSSA. When it comes to gram-negative, cefamycins have improved activity against gram-negatives, especially some of the Enterobacterialis. And cefamycins also have activity against Bacteroides. So that's unique. When you look at cephalosporins, none of them have activity against Bacteroides except for the two cefamycins that we have. Now, third generation cephalosporins, you will see we have the first three are oral, and then we have the IV formulations. So in general, somewhat good activity against gram-positive organisms. And then when you go to gram-negatives, in general, they have pretty good activity against gram-negative organisms, but they are not active against Pseudomonas except ceftazidime. So ceftazidime is the only member of the third generation cephalosporins which has activity against Pseudomonas aeruginosa, and it has a very good activity, ceftazidime. When it comes to fourth generation, cefepime is the only fourth generation we have, and the unique thing about it is against Pseudomonas, excellent Pseudomonas activity, and it also has good gram-positive activity, so beta-hemolytic, strep pneumo, and MSSA. And ceftaroline, which some people would call it fifth generation, and others would call it anti-MRSA cephalosporin, because it's the only cephalosporin in the US with MRSA activity, and it's also has activity against Ephicalus, and good activity against gram negatives, but the only hole in it when it comes to gram negatives is that it does not cover Pseudomonas. So that's one shortcoming of ceftaroline. Ceftobiprole is unique in the sense that it is the only beta-lactam that covers both MRSA and Pseudomonas. So you can kind of think of it as ceftaroline with pseudomonal coverage. And more recently, we had cefiderocol come to the market, and just like aztreonam, cefiderocol does not have activity against any gram-positive, but it has perhaps the best activity against gram-negative. So it pretty much covers anything that's gram-negative that's clinically important, including Pseudomonas and Acinetobacter. Pretty much majority of these beta-lactams are not good against Acinetobacter. Cefiderocol has good activity. So cefiderocol is a novel siderophore cephalosporin. It is still considered a beta-lactam, but so it does bind to penicillin-binding protein, but it's somewhat unique because the way that it enters the cell is that it hijacks the siderophore that comes with the bacteria. So what the siderophores do, they're actually natural iron chelating molecules that are released by bacteria to facilitate the transport of iron into cell for survival. So bacteria do need iron to go into their cell, and what cefiderocol does is that it hijacks. So it's kind of like a Trojan horse, and it hijacks the system to find entry into get entry across the outer membrane so that it can bind to penicillin binding protein. And this is kind of neat because it can overcome some of the mechanisms of resistance in gram negative. So because it doesn't depend on porin channels to enter outer membrane, it doesn't matter if the bacteria make changes in porin channels. And it's also somewhat stable to efflux pumps. So, that's the reason for its excellent activity against many gram-negative organisms. And to make it even better, its structure is stable against hydrolysis by many beta-lactamases, including metallo-beta-lactamases. So remember, when it comes to metallo-beta-lactamases, these are amyler class B. They pretty much break down all of the beta-lactams, and none of our beta-lactamase inhibitors can block these. Now, cefiderocol is actually stable against metallo-beta-lactamases, meaning metallo-beta-lactamases cannot break down cefiderocol. So here's a figure that it shows you how cefiderocol goes across the outer membrane into the periplasm space where it can bind to penicillin binding protein. So essentially it hijacks the system where the bacteria is trying to bring iron in, and cefiderocol hijacks the system and goes in and finds penicillin binding protein. Now let's take a look at the spectrum of activity of carbapenems and cephalosporins. So essentially we have ertapenem, which has a narrower spectrum of activity compared to other carbapenems, and the reason is ertapenem does not have activity against Pseudomonas, so it's somewhat narrower compared to the rest of them. Now all of them are active against Bacteroides, and all of them are active against MSSA, but none of them are active against MRSA. Imipenem has really good activity against pretty much most of the clinically important organisms, including Pseudomonas. It's also active against Ephicalus. with meropenem and doripenem pretty much identical to imipenem, with the exception of Epicalus. They are not active against Epicalus. Now, more recently, we have added beta-lactamase inhibitors to meropenem and imipenem to overcome some of the carbapenemases that are made by Enterobacterales. So you will see that the spectrum of activity in this table looks the same, but essentially the advantage of these two is that, things like E. coli and Klebsiella, if they happen to make carbapenemases, then meropenem and imipenem will no longer be active, and these can potentially overcome those mechanisms of resistance. And in the case of imipenem, because imipenem alone is not stable to AMP-C, if there happens to be an organism such as Pseudomonas that makes AMP-C, then relibactam can block AMP-C so that imipenem will continue to have activity. So when you compare meropenem, verbactam, and imipenem, relibactam, the role of imipenem, relibactam would be that extra activity against Pseudomonas. Otherwise, they both have good activity against carbapenem-resistant Enterobacterales. And lastly, we have cephalosporin combinations with some beta-lactamase inhibitor. So we have ceftolozane, tazobactam. So ceftolozane is kind of similar to cefepime, but it's not available alone. So it was not listed in the previous table. It's only available in combination with tazobactam. So, in general, not much activity for gram-positives, but it has good activity against gram-negatives, particularly for Pseudomonas. So the reason they developed ceftolozane, tazobactam was really to target Pseudomonas that were potentially resistant to other available agents. So if you have Pseudomonas resistant to cefepime and, piperacillin, tazobactam, or even resistant to carbapenems, if you have Pseudomonas resistant to meropenem, ceftolozane, tazobactam could potentially still have activity, so very potent against Pseudomonas. And of course, ceftazidime alone was active against Pseudomonas, and in combination with avibactam, it really has become a potent agent against carbapenem-resistant Enterobacterales, so CRE. So some of those E. coli and Klebsiella that make carbapenemases, ceftazidime, avibactam could potentially be active against those. So, and ceftazidime, avibactam came to the market before we had meropenem, verbactam, and imipenem, relibactam. So it really had a big role in when we had Enterobacterales that were resistant to carbapenems. Ceftazidime, avibactam would have been the only drug of choice at the time. But nowadays we have these carbapenem combinations with beta-lactamase inhibitors. So we have several options now for carbapenem-resistant Enterobacterales. Cefepime and tazobactam essentially has the same spectrum of activity as cefepime. The only addition is that enmetazobactam makes cefepime stable against ESBL. So when it comes to Enterobacterales, so E. coli, Klebsiella, etc., if they have ESBL production, cefepime will be stable to it. So it has improved activity for these gram-negative agents. Now, daptomycin. Let's talk about the mechanism of action of daptomycin. Its class is cyclic lipopeptide. It binds to bacterial membrane in a calcium-dependent manner, followed by the oligomerization of antibiotic molecules within the cell membrane, which results in depolarization, loss of membrane potential, and cell death. So that's the mechanism of action. And when it comes to resistance, it's more likely to be encountered in enterococci than staphylococci. So, for the most part, MRSA is susceptible to daptomycin. It's really hard to find MRSA that would be non-susceptible to daptomycin. It's more common to find enterococci that might be non-susceptible to daptomycin. Now, something cool about daptomycin is the seesaw effect, which essentially means that, as daptomycin MICs go up, in either staphylococci or enterococci, we can see that beta-lactam MIC go down. So it's kind of seesaw. As the MIC of one goes up, the other one goes down. And the mechanism of this effect is not clear, but this has some important roles because if there are situations where we are concerned about resistant development, we could potentially combine beta-lactams and daptomycin, and the combination would be very effective, because if the organisms develops resistant to one, then it becomes more susceptible to the other one, and vice versa. So it's kind of like a seesaw. And this is not just unique to daptomycin, the same effect also applies to vancomycin. So if you were to combine vancomycin with a beta-lactam, there would be a seesaw effect expected, specifically for staphylococci and enterococci. So here's the spectrum of activity of daptomycin. Excellent activity for gram-positives across the board, and absolutely no activity against gram-negative organisms. So this is an antibiotic directed at gram-positives, including MRSA. So excellent activity against MRSA and all sorts of enterococcus, including VRE. So when we have vancomycin-resistant enterococcus, daptomycin would be one of the drugs that would have excellent activity.